Su Majestad la Reina Doña Sofía. Bienvenidos al Congreso Internacional de Enfermedades Neurodegenerativas de la Fundación Reina Sofía y Fundación CIEN. Tiene la palabra el excelentísimo señor alcalde de Elche, don Pablo Ruz Villanueva.
Majestad, honorable vicepresidenta, secretaria general de Innovación, gerente y director científico de la Fundación, ilustrísimo señor presidente de la Diputación, señor subdelegado, autoridades, miembros del Gobierno municipal, miembros de la Corporación municipal, representantes de las entidades de Alzheimer, Parkinson y ELA, ponentes, congresistas, señoras y señores, muy buenos días a todos. Majestad, con profunda emoción y gratitud, en nombre de todos los ilicitanos, quiero darle la bienvenida a Elche y agradecer su presencia en esta importante cumbre internacional de las enfermedades neurodegenerativas. Majestad, esta es su quinta visita a nuestra ciudad. De nuestra familia real, usted, señora, es quien más veces nos ha visitado. Precisamente la última vez que lo hizo fue para reinaugurar, como bien le he comentado, en octubre del año 96, este precioso gran teatro que hoy nos acoge. Su visita y la celebración de este congreso coincide además con el año jubilar Elche, el cielo y la tierra, en el que conmemoramos el 75 aniversario de la declaración del dogma de la Asunción por el Papa Pío XII, eje central de nuestro misterio. La ciudad que hoy la recibe tiene ya cerca de 250.000 habitantes, 3 bienes UNESCO, 9 kilómetros de playa y es una ciudad abierta, con alma emprendedora, laboriosa y socialmente comprometida y leal a España. Y además, por acuerdo municipal, en el año 2013 es una ciudad solidaria con el Alzheimer. Majestad, en el Congreso Internacional sobre el Alzheimer del año 2011 usted afirmó: deberíamos pensar siempre que un enfermo de Alzheimer es alguien que pierde progresivamente distintas capacidades personales, pero que nunca pierde su capacidad humana y sensorial de vivir y de recibir cariño, afecto y cuidados. Como alcalde, pero sobre todo como ilicitano me cabe el orgullo de reconocer, admirar y agradecer que a lo largo de la historia de esta ciudad, diversas entidades y asociaciones han brindado recursos, atenciones, cuidados y esperanza a tantos y tantos enfermos de Alzheimer, Parkinson y ELA. Elche es inmensamente sensible con todas estas personas y cuenta con una nómina ingente de voluntarios y cooperadores que nos recuerdan a lo largo del año el compromiso que Elche tiene y que debemos ensanchar siempre para con los enfermos y sus familias. Por ello, nos llenó de inmensa alegría que la Fundación 100 eligiera a Elche para ser la sede de este importantísimo encuentro. A la Fundación y a su gerente le brindamos una ciudad entera y sin límites y les abrimos las puertas de Elche no solo para estos días, sino para siempre. Porque Elche quiere seguir siendo un aliado absolutamente sincero de esta fundación. Por todo ello, queremos agradecer a su gerente, María Ángeles Pérez, la permanente disponibilidad, su sensibilidad y, tengo que reconocerlo, la amistad que ya hemos tejido entre Elche y la Fundación. Gracias, María Ángeles. Y me van a permitir que como alcalde subraye el orgullo que sentimos en esta tierra bendita porque, además, un ilicitano, Pascual Sánchez Juan, sea el director científico de esta fundación y que desde Madrid, como embajador, con esfuerzo y pasión infunda a su equipo el espíritu que llevamos en el ADN los hijos de esta tierra bendita, los ilicitanos, el espíritu perseverante de una ciudad que nunca se rinde. Quiero trasladar mi felicitación a cuantos dedican su trabajo científico a la investigación sobre las enfermedades neurodegenerativas, cuya prevalencia aumenta de año en año de forma acelerada también como consecuencia del crecimiento de la esperanza de vida. Es decisivo que podamos avanzar en el diagnóstico temprano de estas enfermedades y en el desarrollo de estrategias de prevención que puedan retrasar o detener su aparición. Es decisivo que podamos identificar los factores de riesgo y las causas que las generan. Y es decisivo que podamos encontrar tratamiento eficaz y desarrollar fármacos que puedan frenar, revertir e incluso curar estas patologías. Por ello este no es un congreso más. No puede ser un congreso más, estimados ponentes y queridos congresistas, porque ustedes no pueden olvidar que encarnan la esperanza. La esperanza para nuestra sociedad y para esos familiares que cuando miran a esa persona que les dio el nombre y la vida y ya no encuentran en ellos ni la ilusión en sus ojos ni en el brillo de su mirada, sigue siendo una guía y un motivo por el que luchar. Una neurona que se atrofia o muere en el cerebro de un paciente con una enfermedad neurodegenerativa representa las miles de lágrimas de familiares y amigos envueltos en tantos largos silencios y en infinidad de vidas, de días que se hacen eternos. Con el impulso y el cariño de Su Majestad, la Reina Doña Sofía, con la hospitalidad de todos los ilicitanos, les deseo de corazón lo mejor para estos días y les damos las gracias con toda el alma por llevar a cabo tan importantes investigaciones que son un compromiso de vida. Son ustedes, sin duda alguna, nuestra esperanza. Majestad, ponentes, congresistas, bienvenidos a Elche. Muchas gracias.
Tiene la palabra la señora secretaria general de Investigación, doña Eva Ortega Paino.
Majestad, consellera, alcalde, autoridades, investigadoras e investigadores, colegas, profesionales sanitarios, representantes de todas las asociaciones de pacientes, señoras y señores. Es para mí un honor y un placer, pero sobre todo una gran responsabilidad, dirigirme a vosotros hoy en el marco de este Congreso Internacional de Enfermedades Neurodegenerativas, que reúne a algunas de las voces más destacadas en el ámbito de neurociencia, la medicina y la innovación biomédica. Permitidme expresar, en primer lugar, en nombre del Ministerio de Ciencia, Innovación y Universidades, nuestro más sincero agradecimiento a Su Majestad por su presencia en este acto, pero no solo por su presencia, sino también por toda la dedicación que le hace a través de la Fundación Reina Sofía. Su compromiso con la ciencia y con las causas sociales más sensibles es, sin duda, un ejemplo inspirador para todos nosotros. También me gustaría agradecerle a la organización, a mi querida María Ángeles, porque sé lo difícil que es hacer estas apuestas y organizar un congreso de este estilo, pero también por siempre pensar en mí e invitarme a participar y hacer posible que este espacio de encuentro y reflexión y colaboración sea un hecho. Eventos como este son esenciales para avanzar en el conocimiento, compartir experiencias y, sobre todo, para construir soluciones conjuntas frente a los grandes desafíos de salud que enfrentamos como sociedad. Las enfermedades neurodegenerativas como el Alzheimer, el Parkinson o la esclerosis lateral amiotrófica, las demencias frontotemporales, representan uno de los mayores retos biomédicos y sociales del siglo XXI. Su complejidad clínica, su impacto emocional y económico y su creciente prevalencia en una sociedad cada vez más longeva nos obligan a actuar con urgencia, con rigor y con visión de futuro. Además, su impacto no solo afecta a millones de personas en todo el mundo, sino que también transforma profundamente la vida de las familias, de sus cuidadores y de las comunidades. Por tanto, el abordaje de estas enfermedades exige no solo avances científicos, sino también una mirada ética y humanista que reconozca el sufrimiento, la vulnerabilidad y la necesidad de acompañamiento que conllevan. Estas enfermedades complejas y multifactoriales requieren un enfoque integral que combine investigación básica, desarrollo clínico, innovación tecnológica y políticas públicas centradas en la prevención, el diagnóstico precoz y la atención personalizada. Es por todo esto por lo que la ciencia no puede caminar sola. Necesita del compromiso institucional, de la colaboración internacional y del impulso decidido de todos los actores del ecosistema de I+D+I. Déjenme que les presente nuestra apuesta desde el Ministerio de Ciencia, Innovación y Universidades, ya que estamos plenamente comprometidos con este objetivo. Nuestro compromiso, nuestra acción, se articula en torno a una estrategia integral que refuerza la investigación en neurociencias desde múltiples frentes. Desde luego, apoyando directamente a través del Instituto de Salud Carlos III a la Fundación CIEN y también a través de financiación competitiva del Plan Estatal de Investigación Científica, Técnica y de Innovación, apoyando proyectos que abordan desde la biología molecular del sistema nervioso hasta el desarrollo de nuevas terapias y tecnologías diagnósticas. Además, también respaldamos redes de excelencia como Cibernet, impulsada también por el Instituto, que agrupan a líderes en el estudio de enfermedades neurodegenerativas, promoviendo sinergias entre centros de investigación, hospitales y universidades. Y si han seguido la prensa, no hace mucho también presentamos la nueva iniciativa de Spain Neurotech. Además, fomentamos colaboraciones público-privadas conscientes de que la transferencia de conocimiento y la innovación tecnológica son esenciales para que los avances científicos lleguen a las personas, aquellos que sufren estas patologías. Participamos activamente en programas europeos e iniciativas internacionales que sitúan a España en la vanguardia de la investigación cerebral. Todo ello se complementa con una apuesta decidida por el talento, promoviendo la atracción y retención de jóvenes investigadores, el fortalecimiento de las carreras científicas y la creación de entornos estables y estimulantes para la investigación. Porque creemos firmemente que una ciencia fuerte es la base de una sociedad mucho más justa, más saludable y más preparada para afrontar todos los retos futuros. Dejadme terminar diciendo que la lucha contra las enfermedades neurodegenerativas no es solo una cuestión científica o médica, es ante todo una cuestión de dignidad humana, de cómo queremos cuidar a nuestras personas mayores, a nuestros seres queridos, a quien más nos necesitan. Por eso, desde el Ministerio de Ciencia reafirmamos hoy nuestro compromiso con una investigación que no solo aspire a la excelencia, sino que también esté al servicio de la vida, de la salud y del bienestar de todos. Gracias, compañeros, por vuestro trabajo incansable, por vuestra pasión investigadora y por vuestro compromiso con una causa que nos une y nos interpela a todos. Os deseo un congreso fructífero, inspirador, pero sobre todo lleno de nuevas alianzas. Muchísimas gracias.
Tiene la palabra la honorable señora vicepresidenta primera y consellera de Servicios Sociales, Igualdad y Vivienda, doña Susana Camarero Benítez.
Majestad, excelentísimo alcalde de Elche, gerente de la Fundación CIEN, secretaria general de Investigación, ilustrísimo presidente de la Diputación Provincial de Alicante, señor director científico de la Fundación CIEN, diputados y diputadas nacionales, autonómicas, provinciales, subdelegado del Gobierno en la provincia de Alicante, miembros del Gobierno en Corporación Municipal, representantes de las asociaciones que aquí nos acompañáis, miembros de las fuerzas y cuerpos de seguridad del Estado, investigadores, investigadoras, señoras y señores, muy buenos días. Hoy nos reunimos para hablar de ciencia, de investigación, de futuro, pero sobre todo para hablar de personas, porque tras cada diagnóstico de una enfermedad neurodegenerativa hay una vida, hay una familia, unos recuerdos, unos sueños que merecen ser cuidados con el mayor de los respetos y con todo nuestro cariño. Y quiero empezar con un profundo agradecimiento a quienes hacen posible este congreso. A la Fundación Reina Sofía y a la Fundación CIEN, querida María Ángeles. Vuestra labor es luz en medio de la incertidumbre. Investigáis, acompañáis y cuidáis, pero sobre todo ofrecéis esperanza, lo decía el alcalde, esperanza a tantas familias que sufren por aquellos a quienes más quieren. Y permitidme que hoy ponga en valor algo que me emociona especialmente, el compromiso generoso y constante de Su Majestad la Reina Doña Sofía. En anteriores responsabilidades he tenido la oportunidad, la ocasión, el honor de compartir muchos momentos con Su Majestad y comprobar en primera persona, de primera mano, la entrega, la dedicación, el cariño que siempre demuestra con quienes más nos necesitan. Su presencia no es solo simbólica, es una muestra de un compromiso de toda una vida con la solidaridad, la humanidad y la cercanía. Un compromiso que reconforta, que ayuda y que da esperanza. Muchísimas gracias, Majestad, por recordarnos que la grandeza está en la ternura. Muchísimas gracias. Gracias de corazón. Quienes cuidamos desde las administraciones, desde la investigación, desde las familias, sabemos que las enfermedades neurodegenerativas no afectan solamente al cuerpo o a la memoria, sino a lo más profundo de lo que somos. Y por eso necesitamos acompañar a quienes las padecen. Debemos estar cerca. Cerca con paciencia, con comprensión, con amor infinito. Porque cuidar no es solo tender, cuidar es escuchar, es sostener una mano, es abrazar, es estar presente. Nuestra sociedad tiene una deuda de gratitud inmensa con nuestros mayores. Ellos nos enseñaron a caminar, a hablar, a vivir. Ellos construyeron nuestras calles, nuestras escuelas, nuestras casas. Y ahora nos toca a nosotros cuidarles con la misma dedicación, con la misma ternura con la que ellos cuidaron de nosotros. Nuestro compromiso es no dejar nunca solos a los que más lo necesitan. Y lo demostramos con recursos concretos, pero también con acciones que mejoran la vida de las personas. Lo demostramos con esa atención domiciliaria que permite que nuestros mayores puedan seguir viviendo en su casa, con atención, con cuidado, con seguridad. Pero también con los centros de día que les ofrecen cuidado, compañía, actividades. O con los programas para los cuidadores ofreciéndoles un respiro, formación o apoyo emocional. Porque no olvidemos que la calidad de los servicios también se consigue cuidando del que cuida. También nos comprometemos en ese cuidado de la mano de las asociaciones y de las entidades que hoy estáis aquí presentes, que cada día sostenéis, acompañáis, defendéis a las personas que conviven con estas enfermedades. Desde la Generalitat Valenciana apoyamos su trabajo con ayudas que no solo mantienen proyectos consolidados, sino que también impulsan nuevas iniciativas. Estas asociaciones son el corazón de las redes de apoyo que cuidan de quienes padecen enfermedades neurodegenerativas. Y lo hacen desde la experiencia, pero también desde el cariño y la cercanía. Y esta parte también es muy importante. Son ellas, sois vosotros, quienes nos ayudáis a combatir este mal que, por no estar diagnosticado, no deja de ser igual de importante que el resto, la soledad no deseada. Cada vez más mayores se enfrentan a esa sensación de vacío tan dañina como la propia enfermedad. Y somos conscientes de ello y también trabajamos en combatirlo, con programas sociales de envejecimiento activo, ofreciendo viajes, promoviendo estancias, creando centros, para que nadie, absolutamente nadie, viva sus últimos años sintiéndose solo o abandonado. Desde las administraciones, desde la ciencia, desde la sociedad civil, vamos a seguir trabajando de la mano para ofrecer las mejores condiciones posibles a nuestros mayores y a sus familias. Y en este punto quiero reconocer la labor fundamental de nuestros y de nuestras científicos y científicas y de la ciencia en general, porque gracias a ella avanzamos en prevención, en diagnóstico, en tratamiento. Sois vosotros, los y las científicas, quienes nos ofrecéis la esperanza de encontrar soluciones y mientras llega ese futuro, también nos ayudáis a mejorar el presente de quienes ya conviven con estas enfermedades. Vuestros avances son progreso y orgullo para el conjunto de la sociedad, pero también son alivio y esperanza para quienes las padecen. Para ese abuelo que ya no recuerda cómo se llama su nieta, pero sonríe cada vez que la ve entrar. Para ese esposo o para esa mujer que cuida día y noche de su pareja, pese a que ya no recuerde quién es. Para cualquier persona mayor que cada mañana pregunta por su madre, aunque hace años que ya la perdió. Este congreso está hecho para ellos, está hecho para ellas, para dar voz a quienes a veces no pueden hablar, para dar futuro a quienes temen perderlo, para dar descanso a quienes viven cuidando. Hoy celebramos la ciencia, sí, pero sobre todo celebramos el cuidado, la ternura y la solidaridad. Porque cuando una sociedad cuida a quienes más lo necesitan, se hace más justa, más humana, más digna. Por eso, gracias a todos los que lo hacéis posible. Gracias, Majestad. Gracias a la Fundación Reina Sofía y gracias a la Fundación CIEN. Gracias a todos nuestros científicos, a nuestros profesionales y gracias a todas las personas que cada día cuidan desde el cariño y ponen amor en la enfermedad, porque es lo que más necesitan nuestros seres queridos. Con vuestro ejemplo nos recordáis que cuidar es la forma más hermosa de construir un futuro. Muchísimas gracias.
A continuación, damos paso al vídeo Cerebros Extraordinarios, campaña en apoyo a la investigación científica promovido por la Fundación Reina Sofía.
Hola, soy José Antonio, tataranieto de Santiago Ramón y Cajal, el Premio Nobel de Medicina, el padre de la neurociencia, respetado icono de la generación de sabios. Vamos, un genio. Y vosotros pensaréis, ¿de tal palo, tal astilla? Pues siento decepcionaros. No soy ningún genio, ni soy ningún prodigio. Pero eso no implica que mi cerebro, al igual que el de mi tatarabuelo, no pueda ayudar a descubrir avances extraordinarios para la neurociencia.
Donemos nuestro cerebro a la ciencia. Apoyemos la investigación. fundacionreinasofía.es.
Tiene la palabra el director científico de la Fundación CIEN, doctor Pascual Sánchez Juan.
Majestad, autoridades, colegas, queridos familiares y pacientes. Bueno, lo que hemos visto ahora es muy importante. La donación de cerebros es esencial. Y la mayoría de las charlas, de los avances que vamos a ver a lo largo de hoy se fundamentan en donación de cerebros. Sin los cerebros es muy difícil progresar hacia el conocimiento de los mecanismos de estas enfermedades y en último momento a su cura, ¿no? Por lo tanto, muchísimas gracias a la Fundación Reina Sofía, a Su Majestad, por promover esta campaña que ha sido fantástica, tanto en su ejecución como en sus resultados, porque tenemos constancia, muchísimo impacto, tenemos muchos voluntarios que se han sumado a partir de ella y que estamos muy agradecidos porque esto supone un paso clave, ¿no? Cuando yo estudiaba medicina y ya me gustaban las enfermedades neurodegenerativas, recuerdo que pues disfrutaba mucho con la semiología, la complejidad de estas enfermedades, incluso con la neuropatología, a veces un poco misteriosa, pero bueno, eran enfermedades que realmente ya me resultaban atractivas, muy complejas, pero había una parte siempre que era la más sencilla, que realmente sabíamos que no había que estudiar, que era el tratamiento. Cuando llegabas a la parte de tratamiento de enfermedades neurodegenerativas, pues se decía, pues no hay tratamiento etiológico. Y así quedaba el capítulo. Se hablaba de tratamientos sintomáticos, limitados, pero siempre esto era lo que nos encontrábamos al estudiar estas enfermedades, ¿no? Por suerte esto ha cambiado y el reflejo de que ha cambiado es que hoy estamos aquí dedicando un congreso a avances en terapias en enfermedades neurodegenerativas. Esto es algo impensable, no solo cuando yo estudiaba, sino incluso hace 10 años, que pudiéramos dedicar unas jornadas con ciencia al máximo nivel para hablar de avances en tratamientos. Gracias al proyecto Genoma Humano hemos avanzado muchísimo en mecanismos. Incluso, como nos va a contar el doctor Mojica, ahora somos capaces de manipular el ADN y todas estas herramientas nos están ayudando ya a poder tratar las causas de algunas enfermedades. Primero las más simples, como las genéticas, pero también pronto las enfermedades esporádicas, que son realmente el gran reto al que nos enfrentamos. Y esto está llegando no solo a los laboratorios, sino también a la clínica, a los pacientes. Y por eso estamos muy ilusionados en este congreso donde vamos a mezclar investigación clínica, investigación básica, para intentar llevar al público y a nuestros colegas todo este conocimiento y toda esta esperanza. Y para ello tenemos los mejores ponentes del mundo. La verdad es que estamos muy contentos con la agenda que hemos conseguido, con todos nuestros ponentes, con todo nuestro público. Y nada más, por mi parte, desearles que tengan un congreso muy fructífero y que disfruten de la fantástica ciudad de Elche. Muchas gracias.
Tiene la palabra la presidenta de la Confederación Española de Alzheimer, doña Mariló Almagro Cabrera.
Majestad, autoridades, investigadores e investigadoras, señoras y señores. En primer lugar, quisiera agradecer a los organizadores de este Congreso Internacional de Enfermedades Neurodegenerativas, a la Fundación CIEN, Fundación Reina Sofía, consolidado como foro de referencia en investigación e innovación, la oportunidad que ha brindado a los pacientes y familiares de participar no solo en el encuentro sociosanitario, sino también en este acto inaugural. Ello demuestra, y así queremos entenderlo, el interés y la importancia creciente que desde la investigación se está dando a los sujetos de la misma, es decir, a los pacientes y a los familiares. Porque esto hasta hace poco era impensable, pero ahora lo hemos logrado. Lo estamos haciendo con un nivel de madurez y compromiso con el avance de la investigación, de la que queremos ser actores principales, parte activa de su desarrollo, porque, como podrán imaginarse, somos los principales interesados en que la investigación, en todas sus modalidades, alcance los resultados esperados, que no son otros que la cura del Alzheimer y otras demencias, y así como el resto de las enfermedades neurodegenerativas, que afectan a un porcentaje muy importante y elevado de la población española. A diferencia de otras enfermedades en las que los pacientes pueden adoptar un papel protagonista en sus procesos, en nuestro caso queremos, igual que aquellos, desempeñar un papel activo en todos los pasos o etapas de los proyectos de investigación que se llevan a cabo, desde su concepción, pasando por su diseño hasta su conclusión. Evidentemente, no tenemos los conocimientos científicos ni técnicos que aseguren la investigación que sea de calidad, pero sí podemos y debemos aportar la visión humana de quien no solo ha de participar en la misma, sino de quien va a recibir sus efectos. Porque llevamos más de 20 años sin ninguna solución terapéutica de la que nos podamos beneficiar, si bien en los últimos meses estamos asistiendo a un hilo de esperanza con la aprobación por la Agencia Europea del Medicamento de 2 nuevos fármacos que ralentizan la evolución de la enfermedad y que esperemos estén disponibles pronto en España y de los que puedan beneficiarse personas que de otro modo van a ver comprometida su calidad de vida en un periodo más o menos breve de tiempo. Es lógico pensar que estos nuevos tratamientos están siendo posibles gracias a la investigación en todas sus fases, fase básica, translacional y clínica. Los ensayos clínicos son la herramienta que permiten validar la calidad, la seguridad, la efectividad y la eficacia de nuevos fármacos y, como es lógico, queremos estar presentes en ellos, a pesar de todavía que existen barreras, pero parece que se están disolviendo. Por supuesto, debe entender que no pretendemos inmiscuirnos en la labor de investigadores, pero han de comprender que si los pacientes la investigación no avanza y para que la rueda funcione es imperativo seguir impulsando y aplicando herramientas y tecnologías que posibiliten el diagnóstico temprano, cuanto antes mejor, para poder participar en los ensayos clínicos, pero también para tomar decisiones importantes que van a condicionar la vida y la calidad de vida de las personas afectadas. Esta es la importancia que le damos a la investigación, del interés que tenemos para participar de manera activa y, en definitiva, de sostener la esperanza de alcanzar la cura que haga de las enfermedades neurodegenerativas y, en particular, el Alzheimer y las demencias, lleguen a ser una enfermedad más, con su diagnóstico y un tratamiento tanto cronificante como curativo. En este sentido, y es un orgullo para mí exponerlo en este foro, formar parte del patronato de la Fundación CIEN es un logro para nosotros de trascendental importancia. Para nosotros evidencia la consecución de nuestros objetivos y aspiraciones, pero también la consideración que el propio Centro de Referencia de la Investigación Pública en España tiene de los pacientes y familiares. Para nosotros, la investigación supone el poder cumplir derechos como es el derecho a un diagnóstico y que éste sea precoz, temprano y certero, pero también el derecho a un tratamiento. Y es la investigación la herramienta sobre la que descansa la esperanza de las personas afectadas por las enfermedades neurodegenerativas. Pero también decirles que, como siempre, pueden contar con nosotros. Muchas gracias.
Es el momento de la ponencia inaugural titulada El viaje de CRISPR, rastreando las raíces de una revolucionaria caja de herramientas genéticas. Introduce el doctor Javier Sáez Valero, vicerrector adjunto de Investigación para la Gestión de la Investigación y Transferencia en la Universidad Miguel Hernández de Elche. Expone el doctor Francisco Juan Martinez Mojica, catedrático de Microbiología en la Universidad de Alicante.
Majestat, autoridades, queridísimo alcalde, miembros de las asociaciones, compañeros científicos, conciudadanos, para mí he recibido la encomienda más sencilla y es introducir a mi paisano. Sin duda alguna, todos relacionados con la investigación biomédica conocemos la trascendencia que sus hallazgos en la caracterización de este mecanismo molecular, que fue descubierto, como bien nos presentará, aquí muy cerquita en Santa Pola, han supuesto para el futuro y el presente de la investigación. Esas técnicas las estamos utilizando ya día a día en nuestros laboratorios, pero tenemos esperanza de que aparte las podamos utilizar en lo que es la clínica biomédica. Y, desde luego, como no hace falta presentación, no me voy a extender mucho más, pero es que yo soy también un científico de Alzheimer y no quiero dejar de utilizar esta oportunidad para decir que para mí este congreso va a quedar siempre en el recuerdo. Y lo que perdemos cuando tenemos enfermedades neurodegenerativas es la capacidad para establecer memoria. La memoria la fortalecemos cada vez que estamos recordando una vivencia. Y para mí espero que, igual que yo estoy inmensamente feliz, todos ustedes también puedan recordar este congreso, dado que Su Majestad nos brinda y nos honra con su presencia y su continuo apoyo. La Fundación CIEN y la Fundación Reina Sofía han puesto a nuestra disposición un congreso de una relevancia científica tremenda. Las asociaciones están completamente implicadas y estoy seguro que la charla de Francis va a ser muy amena y nos va a ayudar para que reforcemos ese recuerdo y siempre tengan presente Elche en su corazón. Muchas gracias, Francis. Muchas gracias a todos ustedes por acompañarnos.
Bueno, buenos días, buenas tardes ya. Antes de nada, Majestad, autoridades, señoras y señores, es un verdadero placer estar aquí. Tengo que confesar que cuando Pascual me invitó a impartir esta charla inaugural, inicialmente no me dijiste que era una charla inaugural, me dijiste que era simplemente participar y dar una charla. Le dije que no. Le dije que no porque yo soy microbiólogo, madre mía, y esto asusta, ¿no? Hablar sobre enfermedades neurodegenerativas, digo, ¿dónde voy yo, no? Yo ahí no encajo para nada. Luego me convenció con la ayuda de Javier, dijo, no esperamos que nos hables de enfermedades degenerativas, hombre, lo que queremos es que nos hables de CRISPR. Y ahí sí que puedo empezar a hablar un poco, por lo menos. Eso me convenció, pero no fue lo único. Luego también es cierto, hombre, soy de Elche. Nunca había dado una charla en un congreso en Elche y probablemente nunca más la daré, o sea, era una gran oportunidad. Y la última fue que me comentó que iba a venir Su Majestad, la Reina Sofía, y dije, esta es una fantástica oportunidad, desde luego, para darle las gracias en persona por su compromiso con la ciencia en general, ¿no? y su apoyo a este tipo de eventos. Muchísimas gracias, Majestad. I will switch to English. I prefer Spanish, but anyway. Okay. I think we can manage. Which is the relationship between CRISPR and neurodegenerative diseases? You have a look to the CRISPR-related papers index in PubMed, we'll find that there are many. It's 54,000 at present, papers dealing with CRISPR. And around 1%, 2% of them focus on things, neurology, the different aspects of neurology. This relationship, this connection began in 2014. That is just 1 year after the first CRISPR tool was successfully applied to edit the genome of human cells. And this connection has been very, very fruitful. For instance, with CRISPR researchers are editing and understanding and repairing neurons, rejuvenating neurons. They are also identifying the dysfunctions or protein defects, or for instance, some particular genes that are somehow involved in ALS, autism, or pain insensitivity. Thanks to CRISPR, it is even possible in the near future to move these advances to the clinic, to halt diverse neurodegenerative diseases or to treat them as, for instance, Alzheimer's, which is quite promising indeed nowadays. So, CRISPR technology is really enabling researchers to understand the molecular mechanisms underlying central nervous system pathologies, such as, for instance, epilepsy or autism, including schizophrenia, Huntington's disease, or anxiety. In this way, researchers can also identify potential therapeutic targets to treat them. How do they do that? So one of the ways they can do that is trying to reconstruct the cell lineages of almost any cell during the development of an organism, or to record cellular events. And moreover, with CRISPR, you can identify the genes that are somehow related or involved in specific traits, in specific pathologies. And once you have identified which are those genes, you can edit them to treat the disease, for instance, for gene therapy. You can remove, add, or modify, in general, any sequence almost in any location or within the genome, disrupting this, the function of that gene or correcting unwanted mutations that are responsible for the disease. But this is not all. CRISPR is being harnessed for the regulation and the modulation of gene expression in both directions, activating or repressing their expression, also targeting the transcripts for degradation, or even directly editing the RNA. With CRISPR, you can edit DNA, but also can edit RNA if you don't want to make permanent changes in the genome. So you look at the clinical trials that are now either recruiting or active. There are 100, a little bit more than 100 based on CRISPR. And I just managed to find, probably there are many more, but I just managed to find 4 of them that are related to neurology in general. 2 of them are intended to treat Duchenne muscular dystrophy. Another one to identify molecular targets of depressive disorders, and there's another to evaluate a CRISPR-based RNA editing therapy for MDC. I've been invited here to talk about another thing, the story of the origins of CRISPR, where all this technology comes from. And I'm not going to get into all of those papers, 54,000 papers, but I'm going to cover a long period of time from 1997, when the first repeat called CRISPR was reported in E. coli, to the first CRISPR tool that was developed in 2012, 2013, depending on the opinion of the people. This is a long period of time, but not many contributions, as you can see here, were published. Only a few, so I will try to summarize all these discoveries during that period. Of course, I will emphasize those that I was involved in, and that's, like I said, that's not more. And eventually, I will just summarize just a few advancements and breakthroughs in the last 10 years. So, if you want, I invite you to travel in time, a little bit more than 30 years back in time, to the early 90s. At the time, I was involved in my PhD thesis at the Laboratory of Macrobiology. By the way, the Department of Neurochemistry. We didn't do anything related to medicine in general, because we were interested in something completely different that has nothing to do initially to health or medicine in general. My thesis advisors were Guadalupe Juez Pérez and Francisco Rodríguez Valera. They were some classical microbiologists. They studied microbial ecology. So when I came in to that lab, they told me that they were interested in understanding something very specific. How a microorganism, Halopherax mediterraneae, that was named and isolated by Paco at the time, from the salters, salt marshes in Santa Pola, Alicante, very close from here, was able to adapt, to sense, respond, and adapt to changes in environmental conditions. So the thing is that this guy behaves like Dr. Jekyll and Mr. Hyde. It was crazy for a student when you see one day 12% salts, this kind of colonies on the plates, and on 12% they look completely different, and at least in color. And also when you grow them at 30 degrees, they are very small cells. At 50 degrees, the volume is much larger, still a microbe, of course, but much larger than a 30 degrees. But it's exactly the same organism. Moreover, it's very fast responding to changes in the environment. In just a few minutes, it changes the level and the amount of proteins that are produced in the cell. So trying to understand that, one way to do that was just to modify the genome. First, just try to know what are the letters of the genome, to sequence that. As you probably know, sequencing was very complicated at the time. The first complete genome of a living organism was published in 1995. So we only have a few fragments of the genome of Halopherax mediterraneae available, but we had a genomic library of this organism. I probably don't know what is that, at least in most cases, these plasmids carrying fragments taken from the genome of this organism, discovering all the genome probably. And we knew, for some reasons that I'm not going to get into, that some of those clones probably had genomic information related to the adaptation to salinity. So we started to sequence some of them, and in clone 122, we found this sequence. Probably you're not familiar with that. It's very easy. Just you get, this is the autoradiograph of gel electrophoresis from the products of sequencing. You had to read that manually. From bottom to top, you have to follow the bands. So you have T, C, GG, C, AA, CCC, etc., etc. And reading that, we notice that some sequences, 30 base pair sequences, were repeated 5 times. This was, oh my goodness. These were the first sequencing at the University of Alicante. No one to ask. Most of the time, the sequencing reactions didn't work. And only a few times we got these nice bands that where you can really read a sequence. So we thought probably this is an artifact of the reaction, the sequencing reaction. We noticed that these sequences were partially palindromic. What that means, partially symmetric. So you can read here AACCC, AACCC, the complementary sequence GGTT, which means that this AACC could base pair with GGGTT. You imagine that in space, they can adopt cruciforms, something like a cruciforms due to the formation of intermolecular hybridization. Maybe the polymerase that was used to sequence that DNA, when it reaches this secondary structure, it came back and read it once again, that sequence. And somehow was inventing the sequence in between the repeats, because the repeats were regularly interspaced by unique sequences. Anyway, we repeated the sequence, we came in the other direction and went farther in the clone and with other clones that overlapped those, and we saw that they were real, and they were not 5, but at least 50 repeats. We call this, let's go over there, we call them TREPs, for tandem repeats. And when we sequence the whole clone, we noticed that they was very peculiar, not just because of the presence of the repeats themselves, but there were also other shorter direct repeats and alternating purines and pyrimidines. Look at this, T8, 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 whatever, what's that? So this really looked like DNA structure and DNA topology in this region could be affected by changes in the environment, temperature or salinity, and the supercoiling or the stability of these structures could change depending on those conditions, and that could affect the expression of the nearby genes. Fantastic hypothesis. So in order to check that hypothesis, the next thing to do was to analyze the transcription, the differential transcription, depending on salinity of the growth media, at least, in order to make a connection with halo adaptation to salinity of the genes that we identified there. 2 genes, or 2 open reading frames encoding for proteins, really show that cell-dependent transcription levels. You don't need to understand this, but the intensity of these bands means that higher intensity, higher levels of expression. This RNA would correspond to a protein probably involved in adaptation to high salinity, to 30% salts, and this other probably was involved in adaptation to stressing conditions, both low and high salinity. And what happens with this region here where the repeats were located? They were not in coding regions. No open reading frame, no protein could infer that could be encoded there. But yet we said, let's try Maybe they are expressed, and they are transcribed. But instead of just a single RNA, they produce a population of RNAs that are processed to generate a much more stable RNA molecule. These RNA molecules, small RNA molecules, are now known as CRISPR RNAs or cRNAs. Also, it's important to know that they were not really differentially transcribed depending on the salinity. The transcriptions was not really affected by salinity. Oh my goodness, this is not working good. So the open during frame, the genes next to the repeats have different transcription, but not the repeats. So the hypothesis that they could be involved in the regulation of adaptation, etc., was not so evident. Then we sought to test which could be the role played by these sequences. We didn't dismiss completely initially this role in alloadaptation, and we had to do something. It was, and still is, very difficult to do genetic studies with Halopherax mediterranean in particular. In that time, working with Halopherax volcani, another of these halophilic archaea, was a little bit easier. At least you could introduce some DNA molecules in Halopherax volcani and not in Mediterranean. And we also detected traps, these repeats, in the genome of Halopherax volcani. So we wanted to see some effect on the introduction of extra copies of these repeats in the cell. So introduce a multi-copy plasmid in Halopherax volcani with a few repeats and spacers also present in the chromosome. What we saw initially with these experiments is that the plasmid to chromosome ratio increased when the plasmids carrying these repeats compared to plasmids without the repeats, or empty plasmids. And even more relevant than that, the cells seem to be dying. The viability of these cells decreased a lot when they carried this kind of plasmids. And when we stained the DNA of these cells, we saw a large proportion of poorly stained cells. The cells were losing most of their DNA. This really suggested that traps were somehow involved in an interference mechanism, some interference effect that we couldn't understand at the time. We published those results in a macrobiology journal in 1995. And there we initially just submitted the paper dismissing a few possibilities, trying to explain those results. The first one, the traps were not involved in recombination. Second, they were probably not involved in the regulation of a few genes. Still, we kept proposing that they could adopt these secondary structures, serving as sites for DNA binding proteins to bind there and do whatever they do. Okay. We submitted that, and the editor said, if you want to publish those results in this journal, you will have to propose a role, a function, not just dismiss. So we, being very cautious, we proposed that traps could be involved in replicon segregation. Let me explain this a little bit more. We hypothesized that the traps were something similar to the centromere of the eukaryotic chromosome. So the traps in the chromosome would serve as docking sites for some proteins in the membrane. As the prokaryotic cell elongates, it will drag these chromosomes in order to locate in the middle of the daughter cell one copy of the chromosome before the cell divides. If you introduce a multi-copy plasmid, with the traps, these binding sites to the envelope, they will compete with those of the chromosome. So randomly, some chromosome molecules would segregate in such a way that some cells will become without any chromosome, and the others will get the 2 copies. And the result would be that the cell will die. Everything fit perfectly well with our results, but it was absolutely wrong. As many times happens, that was very disappointing. Just trying to repeat those experiments and trying to do additional experiments to prove that. And we didn't get really anything that supported these conclusions. And as I said before, working with halophilic archaea is quite complicated. But we look at the literature and we got a very nice surprise. In 1987, this guy, a postdoc at a Japanese university, had reported regularly interspace repeats in E. coli. And that's a completely different story. You can work very well with E. coli. You can do almost anything you want. And moreover, in 1991, this Dutch group also reported direct repeats that were very similar, not in sequence, but in the architecture to the traps that we found in halophilic archaea. By the way, they were using them from the very beginning, and this is the first application of CRISPR indeed. They were using them as molecular markers for typing, for the identification of Mycobacterium tuberculosis isolates, because they noticed that different isolates of Mycobacterium tuberculosis carry different sequences as spacers. So just looking at those spacers, you can differentiate isolates. And for instance, TRAC, which is the origin of a tuberculosis outbreak. Then we had 2 more possibilities to work with. Just the fact that these sequences, in the case they were related, they were homologous, and they had a common origin, they have to be very important for prokaryotes, because you look at this phylogenetic tree, prokaryotes are bacteria and archaea. Halophilic archaea is where we found the repeats. E. coli is here, Mycobacterium tuberculosis here. Look at this distance. We are here. And fungi are there, right there, very close. The distance and the genetic difference between these organisms is huge. And these 2 groups of organisms, bacteria and archaea, separated, probably more than 3,000 million years ago. And they were still there. Then we reproduced all the experiments we did with E. coli, and we didn't get any single effect of the introduction of plasmids with the repeats also present in the chromosome. We didn't get any proof of binding of these sequences to the envelope. No DNA binding protein could be isolated and purified. They didn't have any effect at all on DNA topology. They didn't adopt any non-bDNA structure. Really disappointing, desperating, but anyway, have to keep going. And fortunately, after 1995, sequencing was much better than the early 90s. More and more complete genomes of prokaryotes were published, and as they were completed, they were reporting regularly interspace repeats that receive different names. In fact, usually they didn't make any relation with one another. So by 2000, we had already identified this sort of repeats in terms of organization, length, and regular spacing in 20 genomes of bacteria and archaea. Even if you align their sequence, it's possible to infer a consensus sequence, meaning that they are really homologous, they have a common origin. They have to be then very relevant for life, but not for the survival and adaptation at the very different environments inhabited by all of them. So the environment inhabited by each of these organisms is very different. So whatever they were doing, that should be intended to solve some issue that was common to all of them. And no idea what it was. We call them, call these repeats at that time short, regularly spaced repeats. Wonderful, very smart. SR, spacer repeat. But probably I was the only one in the world that thought that was in a nice acronym. The other group that was working with the repeats was the Dutch group, that one that described the repeats in Mycobacterium tuberculosis, that they were still working using them for typing. Then Ruth Jansen, a member of that group, sent me an email telling me that they didn't like at all SR. And he just proposed me to reach an agreement. Sometimes it's better agree than fight. And didn't make much sense if we were just two groups working on the repeats in the world. If we agree on one name, that will be the name that will stay forever. Indeed, I proposed CRISPR for cluster, legally interspace, short palindromic repeats. Awful. I have to confess I don't like it at all, but I proposed several options, and they said CRISPR is just wonderful. It's unique in the PubMed, and it's soundy. Like I said, I disagree with that, but eventually CRISPR was the one that was published, in fact, for the first time in this paper by Jansen and collaborators just a few months later, where they identify 4 genes, and that was the reason, really, why he sent me the email. They wanted to give a name to those genes and proteins, and CRISPR was perfect to say Cas, CRISPR-associated sequences. So they identify 4 genes, Cas genes, Cas 1, 2, 3, and 4. So the CRISPR were not alone for that moment. We knew that CRISPR were at least physically associated to these Cas proteins. They were always very close, in close proximity to the arrays, and were exclusively in genomes with the repeats. So they probably were functionally associated to them, but the function, the real activity of those proteins was unknown. Looking at other proteins that share some homology with them, it was inferred that they should be related to nucleic acids, and that's it. The situation changed, at least for us, in 2003. 2003, we identified CRISPR in all these taxonomic groups of prokaryotes, and we look for sequences matching the spacers of these arrays elsewhere. And we found that a percentage of them were identical, of these spacers, were identical to sequences in mobile genetic elements, viruses and plasmids, but not any plasmid or any virus, invariably in mobile genetic elements of the same species with the corresponding spacer matching sequence. So there was a connection between them, but we were not sure which that connection was. So in order to understand that, we just read about all papers that were dealing with mobile genetic elements, viruses and plasmids, carrying these spacer matching sequences and the relationship with the host, the putative host that was carrying the spacers, the corresponding spacers. And we concluded, after these observations, that CRISPR are constituents of a prokaryotic immune system. We submitted that to Nature. We thought that that was huge, and that probably Nature would be interested in that. They were not at all. And we said, trying to convince them, this will have a tremendous repercussion in biological, biotechnological, and clinical sciences. No way, after 2 mailings with them, it was absolutely rejected, and eventually, 2 years later, we managed to publish the results in the Journal of Molecular Evolution. There we said that CRISPR related to immunity against targeted DNA, that this recognition of this target would be given by the CRISPR RNAs that we identified 10 years before in halophilic archaea. And in other words, that CRISPR-Cas is an RNA-guided acquired immunity system. And moreover, because just spacers match sequences, genomic sequences, not related to mobility, we also suggested that they could be involved in some more versatile regulatory system, as it was proven years later. So, prokaryotes do not have brain. They don't have a nervous system. They don't have sensory organs, of course. But they can, and it was known at the time, they can sense changes in the environment, respond to them, and adapt to them. And some say that it could be considered that at least some prokaryotes have a nose, and they detect these changes in the environment, and they move. If it's a nice perfume, they go there. It's awful perfume, they just go back. It was already well known that even though prokaryotes lack speech, they are capable of communicating one another. And it was already known also that prokaryotes can defend themselves from invaders, mainly viruses. But this was really unexpected, is that prokaryotes can remember. They have some kind of memory. They can record infections, and descendants of those that recorded that infection will recognize that virus, a descendant of that virus, and somehow neutralize it. This was really, really unexpected. This discovery marked a turning point, stimulating not many, but just a few macrobiologists and biochemists to investigate in the mechanism of this unique defense system. As you see, not many papers during all this time. These papers were just complete genomes where they reported similar repeats, and that's it. But that changed it after 2005. It changed to the point that NASA, for instance, was interested in these CRISPRs, and the first CRISPR meeting was a mini meeting, was held in Berkeley in 2008. Mini meeting indeed, 10 speakers, only 10 speakers. Only 4 of us have published on CRISPR. Kira Makarova, for instance, who took our message that this CRISPR could be something similar to the eukaryotic RNAi, the interference RNA system of eukaryotes, and they proposed an hypothetical mechanism of action. Just 1 year after we published this paper, Rudolf Barangou, who showed that when you challenge a bacterium that is sensitive to viruses with one of those viruses, sometimes they take pieces of the DNA of the virus and integrate them in the CRISPR array. And after that, the bacterium is resistant to infection. John van der Oost was also there. He had a paper already accepted for publication in Science where he confirmed that the CRISPR RNA molecules serve as the guides of the system. The other was myself. I presented there what I call the PAM, Protospacer Adjacent Motif. That is a motif, a conserved sequence, very short, from 2 to 5 nucleotides that is conserved next to the protospacers. Protospacers are the sequences from which the spacers derive. And we noticed that they can change depending on the CRISPR-Cas system and organism, these motifs, and we propose that they should be important for the recognition of those sequences as targets. PAMs will have to be considered in the development of the expected applications of the CRISPR system as an innovative molecular biology tool. No molecular biology tool had been developed at the time, but it was expected. And it was not developed so far because we didn't know for sure how the virus or the plasmid is neutralized by the CRISPR system. We knew how it's recognized, but no how it's neutralized. Either just binding there to the DNA, to the target, cleaving, whatever. And the response was given by Sylvie Moineau, who said or shown that the CRISPR system, at least one of the CRISPR systems that were already known at the time, recognized the target and clips there, just there, within the target. And the only thing you need to do that is to have a PAM motif next to it. Only when that motif is next to the spacer matching sequence, the machinery will cut within the spacer and destroy it. In 2011, we already knew that there is not just one CRISPR system. There are many CRISPR-Cas systems. At the time, we knew that there were 3 types of CRISPR-Cas systems and a few subtypes. And model organisms carrying some of these CRISPR-Cas systems were studied by some of these researchers, and they were partially characterized. So this allowed us to depict a general mechanism of action with the few things we knew, with 3 stages: adaptation, expression, and interference. The first one seemed to be common to all of them. When a virus enters one bacterial cell, the Cas1 and Cas2 proteins take pieces of the DNA and integrate them into the CRISPR array. After that, the CRISPR array is transcribed and processed to generate the CRISPR RNA molecules, each of them carrying a single spacer, sequence from a single spacer. These CRISPR RNAs will guide proteins to recognize and cleave the target. But there was something missing in order to develop a CRISPR tool. It was not known which were the components of any of these CRISPR systems that were absolutely necessary and sufficient to carry out this interference, the recognition and cleavage. That happened in 2012, just 1 year after this first proposal of CRISPR classification. These 2 papers were published in 2010, where they identified Cas9, that is the a protein associated to one of these types of CRISPR-Cas systems, the CRISPR RNA, the guide, with a spacer sequence that allows the recognition of a matching sequence to produce the cleavage, and a third component, the tracer RNA. It's an RNA that is encoded next to the CRISPR loci that was also necessary for that. This group failed to identify tracer as a necessary element of this tool. So if you want to cleave any sequence of DNA within a cell or in the test tube, the only thing you have to do is add Cas9, add tracer, and a guide that you can synthesize or engineer or make the cell to produce it. You cannot change this sequence, but you can change this. As far as it matches where you want to produce the cleavage, Cas9 will go there and will cleave the two strands of the target, provided that there is the right band there. So you can cleave any sequence, almost any sequence, given that there is a band sequence next to it. So at the end of this paper, they said that this could be exploited for RNA programmable genome editing. And they got the Nobel Prize in Chemistry 8 years later for that. Notice that it was given that for the development of a method, not the method. 1 year later, two groups led by George Arch and Feng Zhang in States, they showed that the system, this tool worked perfectly well to edit the genome of human cells. This was the first application, indeed, of this CRISPR tool. Why is not the method, but just one method? Because there were other methods for genome editing. People was using finger nucleases and tailing, adios, here, and tailing to edit the genome of eukaryotic cells, for instance. It was based in the fact that you can cleave DNA within the cell. You program these proteins to cleave, to go to one given place with a given sequence and to cleave within that sequence, producing a double-strand break, and then our repair systems will try to repair that. And depending on the system, you will get an output or another. So one of them just remove or will add some nucleotides, and the consequence of that usually is a disruption of the function of this gene. And the other one, which is much more interesting, is the homology-directed repair system. You can provide the cell with this tool and also with a template, a DNA template, that shares some sequence in common with that sequence that is both sides of the place where you produce that cleavage. And in the middle, you can introduce whatever you like. So you can change the information at that place. What happened then that explains that CRISPR was so popular after that moment. What's better for CRISPR than for the others? Many reasons. The most important probably is that it is easily programmable. It's very easy to program it. Another reason is that it's very easy to use in general. Also, it's very accurate, very precise, and even more important, very cheap. It's quite cheap. You can get CRISPR from yogurt, Streptococcus thermophilus, one of the bacterium in the yogurt. You have the millions of copies of the tool. You can purify them quite easily. You can program this protein to cleave any place with just a few euros. That is the cost of synthesizing the DNA from which you can get the guide RNAs. So that explains why many people that were using zinc fingers or tailing for genome editing moved to CRISPR. And what is much more important, researchers that never thought about the possibility of doing genome editing just adopted CRISPR in their laboratories. All of this is Cas9. Cas9 is just one protein of the type 2 systems of E. coli. We know now that there are 8 types of CRISPR-Cas systems, over 30 subtypes, and more than 100 variants of CRISPR-Cas systems. And they differ in the identity of the Cas proteins. Some, instead of Cas9, have Cas12, Cas13, Cas14, Cas10, Cas3, that are the responsible for the recognition and cleavage of the target. So you have a bunch of proteins that can be used instead of Cas9. So also they differ in the mechanism they use in a few features and in some cases. So we can't talk about the mechanism of CRISPR, but the mechanisms of CRISPR, many mechanisms that could vary substantially. For instance, some systems take spacers from RNA in addition to from DNA. Some systems cleave RNA instead of DNA. Even though the vast majority of CRISPR-Cas systems so far characterized share the fact that they are guided by these guide RNAs and recognize specific sequences, some of them cleave that sequence, others just bind there. And the consequence is that instead of immunity against that mobile genetic elements, they play regulatory roles. In this case, for instance, is a pathogenic bacteria that, thanks to CRISPR, is now recognized by the immune system of the host. Recently, CRISPR systems associated to transposons have been discovered. These are CRISPR-Cas systems that guide, you know what our transposons are, jumping DNA that moves to one place to another and integrates almost randomly. In those cases, CRISPR tells the transposase where it has to introduce that transposon. That's very useful for genome editing, for integration of large fragments. And moreover, some systems, after recognizing the specific target, they became crazy. And they start cleaving any DNA, any RNA within the cell. That's called collateral cleavage. So not just a tool, not just CRISPR-Cas9, not just CRISPR genome editing, but we have a huge CRISPR toolbox. Based on cleavage, you can kill bacteria. The same system that defends bacteria from infection by viruses can be used as a sequence-specific antimicrobial to kill exclusively Clostridium tetani, carrying the tetanus toxin, and that's it, okay? You can program one of these tools to kill only bacteria that carry a particular trait, a particular sequence. Also, based on the collateral cleavage, molecular diagnostics devices have been implemented, very cheap, very sensitive, very precise. Apart from cleavage, either native or engineered Cas proteins have been also used, for instance, as carriers, not just to cleave, not just as nucleases, but as carriers of any activity. You can fuse this dead Cas protein with any protein, any domain, any activity, and easily take it to wherever you want in the DNA. And with that, you can activate or repress transcription with transcriptional activators or repressors. You can modify, epigenetically modify the DNA, methylate the DNA, acetylate histones with an epigenetic modifier. You can illuminate as a rainbow. In fact, it's called the rainbow, CRISPR rainbow. You can illuminate the several loci within the cell in the DNA at the same time and study how it behaves. You can change the location of the chromosomes in the nucleus move them to one place to another, and change the architecture of those fragments of DNA. And even more important, you can fuse Cas9, the dead Cas9, with deaminase, adenine deaminase, for instance. And you perform a chemical modification of the base, and you convert it into another base. Editing without cleavage. Many diseases are due to the change of one base for another. You can revert that. And one of the applications that I like the most is recording. It's based on the first stage of the CRISPR mechanism, the adaptation. As I mentioned at the very beginning, CRISPR has been harnessed to trace cellular events, okay? For instance, detecting RNA that is produced in the cell and is integrated in the CRISPR array, and this is synthetic spaces from RNA. You can also trace cell lineages, but in general, you can record any information and store any information in a CRISPR array. And any information means any information, as far as you can re-encode that information using the 4 letters of the DNA. And that's the case of a movie. So this is a movie. It's not a wonderful movie, just a collection of photographs. The left is the original image, and the right is the image reconstructed after introducing that information, pixels encode using combinations of the 4 letters of the DNA, pieces, fragments of DNA that the E. coli introduces in the CRISPR array. You recover that after a while, and you can decode that information, and you can get any information stored in the CRISPR arrays. This is really amazing. And that's my point of view. But what I really think is truly remarkable is the fact that prokaryotes are capable of learning. To me, that was really astonishing. They can remember fragments of DNA that an ancestor obtained from an invader generations earlier, and use that memory to destroy it. They can evolve for survival just thanks to that sort of memory. Probably you know the evolutionary theory of Darwin. Evolutionary theory is more than a theory, of course. Every single living being evolves thanks to the selection of the fittest that was generated by spontaneous and random mutations. Prokaryotes do that, but they also have some intention. They can also evolve, contribute to that diversity, taking pieces, knowing what they are doing, okay? So this is really unexpected it was, and it's really nice that from this knowledge that happens sometimes, most of the time, all these useful tools have been implemented. So just to finish, I think I'm more or less on time, I would like to thank my collaborators during all these years, in particular my scientific parents, Guadalupe and Paco, who showed me the way I follow all this time, the basic and fundamental research. And I would like also thank those institutions that still support us with funding for our fundamental research. And thanks also to all of you for your attention and for looking after our hard drive. Thank you so much. Thank you.
De este modo damos por finalizado el acto inaugural del Congreso Internacional de Enfermedades Neurodegenerativas. Muchas gracias.